Hello everyone, Assalamu alaikum. In this video lecture, I am going to discuss with you about the medicinal chemistry of NSAIDs. Mechanism of action of NSAIDs. NSAIDs are one of the important class of the medicine that act by inhibiting the cyclooxygenase enzyme. Cyclooxygenase enzyme, that is also known as the COX enzyme, has the important role in the synthesis of the cyclic endoperoxides. The cyclic endoperoxides are basically formed from the arachidonic acid. So ultimately they are involved in the synthesis of prostaglandin. There are the different type of prostaglandin that are present in the human body. So on that basis, there are the main two cyclooxygenase enzymes that are catalyzing their functions. One is the COX-1 and other is the COX-2 enzyme. So they are differing from one another on the basis of their function, on the basis of their selectivity for the particular receptor inside the body for the different organ. So like COX-1, that is the cyclooxygenase 1 enzyme, has the important role in those prostaglandin that are involved for maintaining the normal physiological activities of the different cells of the body. Like those cells that are involved in the protection of the gastric mucosa, means the stomach lining, and some are involved in the maintenance of the kidney functions. On the other hand, COX-2 or the cyclooxygenase 2 enzyme is responsible for the production of those prostaglandin that have the important role in the inflammation and inside the body, means they are involved in the inflammatory responses. So mostly the NSAIDs inhibit the both COX-1 and COX-2 enzyme, but these are the non-selective NSAIDs. So depending on their variety of selectivity, they can be classified as selective and non-selective. So the non-selective, those who are inhibiting both COX-1 and COX-2, may ultimately have the different side effects. So for that purpose, selective COX-2 inhibitors are used. So selective COX inhibitors are used in order to eliminate the side effects that are associated with the NSAIDs due to the COX-1 inhibition, because the COX-1 inhibition may causes the side effects that are related to the gastric and the renal effects. Classification of NSAIDs. This classification of NSAID drug is based on the chemical structure of their drug class. Like some of NSAIDs are the salicylic acid derivative, means their basic ring nucleus, or what we say the parent drug nucleus, consists of the salicylic acid. The examples of drugs are aspirin, diflunisal, salicylate, and sulfasalazine. So all these drugs have the basic ring nucleus, that is the salicylic acid. The second class is the para-aminophenol derivative. The well-known drug that is the paracetamol belongs to the class of para-aminophenol and the phenacetin. Third class is the pyrazolidine dione derivative. So pyrazolidine dione means they have the chemical structure in which the pyrazolidine ring is present, and dione is representing the 2 ketone functional group. The example of drug that belongs to this category are the phenylbutazone, oxyphenylbutazone, and the sulfenpyrazone. And other class is the Anthranilic acid derivative. They contain the Anthranilic acid ring nucleus. The example of drug is the mefenamic acid, flufenamic acid, and the meclofenamate. There is another important class of the NSAID that contain the aryl alkanoic acid derivative. They are differing from one another on the basis of their side chain. Like some have the indole acetic acid, that is the example of drug is the indomethacin, andine acetic acid, that is the sulindic, pyrrole acetic acid, that is the tolmetin, and the phenyl acetic acid are the propionic acidic derivative. So the among the aryl alkanoic acid derivative that contain the phenyl acetic acid are propionic acid ring. The example are the ibuprofen, that is the ibuprofen syrup, that contain the ibuprofen, and the ibuprofen tablets, the diclofenac, naproxen. So all these are belong to the aryl alkylonic acid derivative, but that contain the side chain, that is the phenyl acetic acid or propionic acid. There is another class that is the oxychem derivative. Oxychem have the different drugs. Example are the pyroxychem, meloxicam, and the tenoxicam. Besides this, the selective COX-2 inhibitor means they only inhibit the COX cyclooxygenase 2 enzyme. The example of the drug are the Salicylate, rofecoxib, and the valdecoxib. Another class is the gold compound that contain the gold element in their chemical structure, metal in their chemical structure. So the example of the drug is the aronofen and the arothioglucose and the arothioglucamide. Miscellaneous NSAIDs include the nebumetone and the nimesulide. Beside this, there are other different NSAIDs that can be useful in the gout treatment. The example are the allopurinol, probenecid, and the sulfenpyrazone. So now let's discuss them one by one. First of all, the salicylates. Salicylates are those class of NSAID that do not only possess only the antipyretic and analgesic, but they also have the anti-inflammatory function. But beside these, they have proven to be therapeutically more useful in the excretion of uric acid. So as they are going to be enhancing the excretion of uric acid, so they are thought to be useful in the treatment of the gouty arthritis. The more attention have been given to this class because of their ability to inhibit the platelet aggregation. Means they have the important role in the inhibition of the platelet aggregation. So that's why they contribute to the heart attack and the stroke, and useful in the reduction of the risk of the myocardial infarction because of their ability to inhibit the platelet aggregation. In addition, a recent study suggested that the aspirin and the other NSAIDs that belongs to the salicylates class can have the protective role in the colon cancer. Let's check out the structure activity relationship of the salicylic acid or the salicylates. So this is the chemical structure of the acetyl salicylate. You may observe that this is the benzene ring, and this is the carboxylic acid functional group, and this is the acetyl functional group. So the chemimal name or the IUPAC name can be the 2-acetoxy. This is the 2-acetoxy benzoic acid or the acetyl salicylate. We can also simply termed as the acetyl salicylate, or this is the 2-acetoxy because this is acetoxy or ester functional group benzoic acid. How this is synthesized? Let's check out the synthesis of the aspirin. This aspirin can be synthesized simply by the reaction of salicylic acid with the acetic anhydride. So in this reaction, you may observe that over here at the second position, hydroxyl functional group is being acetylated. So this is how aspirin is formed. The active moiety, or what we say the main pharmacophore in the chemical structure of salicylate, is the salicylate ion, means salicylates ion, and all the sites of aspirin that are particularly related with the GIT effect are associated with this carboxylic acid functional group. So in their derivative can be synthesized by reducing the acidity of this carboxylic acid functional group. So this will ultimately affect the potency of the drug. There are the different type of amide derivative, which we have already studied that these are the kind of the products. So amide derivative of salicylic acid can be formed, that is the salicylamide. The difference is that the salicylamide there can retain the analgesic action of salicylic acid, but it does not have the anti-inflammatory property. So if we are going to form the amide derivative of salicylic acid, it can have the analgesic action, but it cannot have the anti-inflammatory property. Next, we can also substitute the carboxylic or phenolic hydroxyl functional group, but this will again ultimately affect the potency. This will again affect its potency. Next is the replacement of the phenolic hydroxyl, the hydroxyl functional group that was present in the phenolic. If we are going to replace its meta or para position in relation to the carboxylic functional group, this will abolish or what we say destroys the activity of the aspirin. So let's check out this one. This is the hydroxyl functional group. If we are going to substitute it to the para position, or what we say the meta position, in relation to this carboxylic acid functional group, so this will ultimately destroys the activity. Placement of phenolic hydroxyl group. And another is the, if we are going to place the another additional hydroxyl functional group at this position or at this position, this will again ultimately destroys the activity of the aspirin. Next is the substitution of halogen atom on the aromatic ring. If we are going to substitute the halogen, that are the electronegative or electron withdrawing group, in the aromatic ring, this will increases the potency, but at the same time it will also increases the chances of the toxicity. If we are going to substitute the aromatic ring at the 5th position of salicylic acid, means if we are adding the aromatic or phenyl ring at the 5th position of salicylic acid, the anti-inflammatory activity of the drug can be increased. The example of this derivative drug is the diflunisal. This is the structure of the diflunisal. You may observe that it has the additional additional phenyl or aromatic ring at the 5th position, and which is further substituted with the 2-fluorine. So its IUPAC name is the 5, means this is representing 1, 2, 3, 4, 5, the 5th position, 2,4-difluorophenyl salicylic acid. So this is the salicylic acid derivative. So if we are going to substitute the salicylic acid at its 5th position with an additional aromatic ring, so this will increases their anti-inflammatory effect. Let's check out the metabolism of the salicylic acid derivative. The initial basic route of the metabolism of these derivatives to conversion into the salicylic acid, means first salicylate ion must be converted into the hydroxyl, and so ultimately salicylic acid will be formed. So that will be excreted as a free acid 10% or undergo the conjugation. So major route of the conjugation is with the glycine. Glycine conjugate is the major metabolite for the salicylic acid, that is the 75%, and almost 15% will be formed with the conjugate with the glucuronide, that is the glucuronic acid. A small amount of metabolite that can be also a result of the aromatic hydroxylation, means that the aromatic ring hydroxylation can take place, and that will lead to the other inactive derivative, that is the geneticic acid. Now let's discuss the next class of the NSAIDs, that is the para-aminophenol derivative. Para-aminophenol derivative are those classes that possess the analgesic activity, antipyretic activity, but they lack the anti-inflammatory effect. So among the para-aminophenol, there are the different drugs that were the previously reported, like acetanilide, phenacetin, but they are being banned, or their use has been stopped because of their highly toxic nature. Like let's discuss the acetanilide. This was introduced in the therapy in the 1886 as an antipyretic and analgesic agent. This was used as an antipyretic. It has role in the fever, and the analgesic means the pain relieving, but it was later on it was found to be highly toxic, means there are the severe toxicity, that is the methemoglobinemia and the jaundice were result from the use of the acetanilide. So there was another class of the para-aminophenol derivative drug, that is the phenacetin. Phenacetin was introduced in the following year, this was introduced in the 1887, and this was also widely used as an analgesic antipyretic, but it was also, its use was also withdrawn because of its severe toxicity on the kidney, that is the nephrotoxicity. The only well-known derivative that is being widely used till today is the acetaminophen. Acetaminophen, that is simply the paracetamol. Paracetamol was introduced in the 1893, and it remained the only useful group, and its main therapeutic use is the antipyretic and as an analgesic agent. So acetaminophen paracetamol, that is the Panadol tablet which we are taking, is basically belong to the chemical class of the para-aminophenol derivative. So these are the chemical structure. You may observe that this is the acetanilide structure, this is the acetanilide, and this is the chemical structure of phenacetin that contain the ethyl, and this is the paracetamol chemical structure. Metabolism of the para-aminophenol derivative. Metabolism of para-aminophenol, they undergo mainly through the hydrolysis. Their hydrolysis will take place, and after hydrolysis they will be converted into the aniline derivative. Aniline derivative can form again the different type of amine that are the hydroxylamine, such as acetaminophen, for example, going to the first pass metabolism, and then when converted in hydroxylamine, and then will form the conjugate with the sulfate, that are the orthosulfate conjugate. The N-hydroxylamine which is formed can be converted into the more reactive toxic metabolite, that is the N-acetaminophenol. N-acetaminophenol is the toxic metabolite that is formed as a result of metabolism of the paracetamol, and this has produced toxicity to the different organs that are the kidney, liver, in conjugation with the hepatic glutathione to form the mercapturic acid and the cysteine conjugate. So this will again can form the hepatic glutathione conjugate with the glutathione and glutathione, which will result into the compound like mercapturic acid and the cysteine. SAR of para-aminophenol derivative. Like first of all, let's get the etherification of phenol. If we are going to convert the phenol hydroxyl into the ether, the functional methyl or propyl ether derivative were reported to have the greater side effect than the ethyl derivative. So the stable, or what we say the highly potent or therapeutic useful ether, were formed only when we are substituting with the ethyl. So the example is the phenacetin, that is the para-ethoxyacetanilide. Clear. On the other hand, the methyl or propyl ether were not that much therapeutic useful. Later on, it was also observed that phenacetin has the nephrotoxicity, and it was also replaced by the paracetamol. Substitution of nitrogen atom. If we are going to substitute the nitrogen atom, then it may reduce the basicity, but also reduce the activity. So activity unless the metabolic labile. So nitrogen atom can only be substituted if it is going to be converted into that derivative or metabolite that can be easily undergo through the metabolism, means that can be metabolically labile. For example, the group that is the acetyl group. Amide derivative. Amides, those are derived from the aromatic acid like the N-phenyl benzamides, are the less active or inactive. So if we are going to form the amide from these aromatic acids, the derivative are the N-phenyl benzamide, ultimately they will not have any therapeutic role. The next class is the 3,5-pyrazolidine dione. 3,5-pyrazolidine dione. The example of the drug in this class are the phenylbutazone, oxyphenylbutazone, sulfenpyrazone. So this is basically the pyrazolidine ring that contain the 2 nitrogen atom, and it is the 5-member ring. But 3,5 is representing the position of the 2 ketone group, that is the dione. This is the 1, 2, 3, 4, 5. So the and beside this, the both of the nitrogen are being substituted with the 2 aryl side chain, and this R and R1 is representing the different type of functional group or substituent that are present in different. For example, in case of phenylbutazone, we have the hydrogen over here and the butyl side chain at this position. In case of oxyphenylbutazone, oxy itself is representing, means it is the hydroxyl functional group at this position and the same butyl side chain at this position. Sulfine pyrazone is basically representing the hydrogen at this, and the side chain that is present at this is the contain the sulfur double bond with the oxygen, that is sulfone group. So now let's discuss the SAR of the 3,5-pyrazolidine diol. If we are going to substitute the nitrogen, one of the nitrogen atom, which nitrogen? This one. If we are going to substitute with the oxygen, that will form again the isosteric derivative, that is the isoxazole analog. So these isoxazole analog are also as reactive as the pyrazolidine derivative. In other case, in the 3,5-pyrazolidine, the pharmacological activity is mainly because of the dicarbonyl derivative. These dicarbonyl derivatives are basically essential in maintaining the acidic characteristic of drug. That is because of the proton, this hydrogen that is present at the 4th position. So the presence of keto group in the gamma position in the butyl side chain can produce the active compound. So for example, in the at this side chain, like this side chain is present is here, if we are introducing the ketone functional group in this side chain, it will again result in the derivative that have the activity, decreasing or eliminating the acidity. If we are, what happen if we are going to remove the acidic proton? If we are going to remove the acidic proton at the 4th position, that is the 4,4-dialkyl derivative, this will cause the abolishes or completely eliminate the anti-inflammatory effect. So if this 4th position hydrogen is in proton is important in maintaining their anti-inflammatory characteristics. For and the other end, if acidity is enhanced too much, means if more proton are added or any other substituent that can enhance its acidity, then its anti-inflammatory and sodium activity can be decreased because of the increase in the limit of the acidic characteristic. So on the other hand, but their properties like uric effect can be increases. Uric is their ability to create out the uric acid. The next is the introduction of polar functional group. If we are introducing the polar functional group at the 4th position in the side chain, they will give the mixed result. So this will again depend on the which type of polar functional group. For example, the gamma hydroxy and butyl derivative have the good uric activity, but their anti-inflammatory activity will be less. And on the other hand, if we are going to substitute the 2-phenyl thioethyl group at the 4th position, that will result into the compound that will have the good anti-gout activity, means that will the high uric effect. So the example of the drug is the sulfine pyrazone. Sulfine pyrazone have the 2-phenyl thioethyl side chain at the 4th position. Presence of the both phenyl ring is essential for their is not essential for those activity, but their activity was being varied only because of this 4th position side chain. Clear? So this position is very much important rather than the side chain. Meta substitution of aryl ring. If we are going to substitute the aryl ring with the at the meta position, this will result in the phenylbutazone give uniformly inactive compound. Para substitution such as methyl, chloro, nitro or hydroxyl can retain C activity. So the meta substitution of aryl ring will decreases their activity, or what we say the results in the compound that are inactive, and but para position with the functional group like methyl, chloro, nitro or hydroxyl can retain their activity. So these are the again the basic ring skeleton and the phenylbutazone, oxyphenylbutazone, sulfine pyrazone. So these structure can be asked in the exam. You to draw, depending upon the question. They draw the structure of phenylbutazone, oxyphenylbutazone. You have to just replace these functional group at this R and R1. Next drug is the phenylbutazone. Phenylbutazone, its trade name are the Pyrazolidine and the Busone. This is the chemical structure of the phenylbutazone. You may observe that is the Pyrazolidine ring at the 3rd and 5th position to ketone, and this is the what we say the isobutyl n-butyl side chain that is present at the 4th position. So its IUPAC name is the 4-n-butyl 1,2-biphenyl 1,2-biphenyl 3,5-pyrazolidine dione. Its physical properties, it acts as a white crystalline powder, practically insoluble in water but sparingly soluble in alcohol and soluble in the alkaline solution. It is the pyrazolidine derivative, and its the therapeutic properties are the antipyretic, analgesic and anti-inflammatory action. But as this was not frequently used because of its toxicity, it is used it was useful earlier in the treatment of the osteoarthritis and the ankylospondylitis arthritis and the superficial thrombophlebitis and the painful shoulder diseases, because in these cases the less toxic drugs have the failed. The next class is the Anthranilic acid derivative, that is the phenamates. The phenamates is the name given to the Anthranilic acid class of the drug. The Anthranilic acid NSAIDs result from the application of the classic medicinal chemistry, that is the biosteric drug application concepts. This is the Anthranilic acid ring nucleus chemical structure. You simply draw the two benzene, connect them by the NH, and just place the benzoic acid or carboxylic acid at the upper benzene. The SAR of the phenamates are the Anthranilic acid. The position of carboxylic functional group is important. This one is important. And if we are going to 3 and 4 amino benzoic acid, then they will not have the activity. So this is indicating that if we are going to substitute the amino functional at the 3rd or 4th position, or 3 or 4 amino benzoic acid derivative, if we are going to convert it, their activity will be abolished. A replacement of carboxylic acid functional group with the isosteric tetrazole means the tetrazole is one of the important heterocyclic ring. If we are going to substitute this, it will result in the compound that will have again the good anti-inflammatory activity. If we are going to substitute the placement on the Anthranilic acid ring, generally reduces the activity. Means if we are going to substitute the different functional group at the Anthranilic acid NH group, it will causes the reduction of activity. Like substitution on the N-aryl ring can lead to the different result, like these results were observed in the two different biological assays. One assay was the ultraviolet erythema assay for the anti-inflammatory activity. In that case, the order of activity was the tertiary was greater than secondary, and that was greater than the quaternary. For the mono substitution with the CF3 group that is present in the flufenamic acid drug. On the other hand, in case of the red paw edema assay, that was observed that the 2-chloro derivative were the more potent than the 3-chloro. Clear? So and these were the two different results observed in the two biological assay. In case of the di-substituted derivative, means the derivative that have the two substitution at the two different position, like in case of the 2,3 di substitution, that is the present in the case of the mefenamic acid, the derivative will have the more potency or the more therapeutic efficacy. The NH moiety of the Anthranilic acid is essential Like for example, if we are going to substitute this NH with the oxygen, methyl, sulfur, sulfone functional group or any other functionality, it will cause the reduction of their activity. This is the chemical structure of mefenamic acid. You may observe that this is the Anthranilic acid ring nucleus, but this benzene ring is being substituted with the two additional methyl group at the second and third position, and due to these additional 2,3-dimethyl side chain, they have the higher efficacy, therapeutic efficacy in comparison to the other Anthranilic acid derivative. An analog approach, this kind of derivative can be synthesized by the reaction of the orthochlorobenzoic acid. Means this is the indicating the synthesis of the mefenamic acid. So if I will ask you in the exam, how will you synthesize mefenamic acid, you have to just mention this statement, that by the reaction of orthochlorobenzoic acid with the 2,3-dimethyl aniline, ultimately this will result in the compound which has the structure like this, that is the mefenamic acid. As far as its metabolism is concerned, its metabolism mainly occurs at the oxidation at the third position methyl group, and then it will undergo through the conjugation with the glucuron acid, and the resulted derivative will be the mefenamic acid. The majority of 3-hydroxy methyl metabolite and the dicarboxylic acid product are produced, and that are the inactive, and they will be ultimately excreted out. As far as their therapeutic use is concerned, they are mainly used as an analgesic and the anti-inflammatory. The next class of these drugs are the aryl or the heteroaryl acetic or propionic acid derivative. Among them, the chemical structure of the ibuprofen and the diclofenac are only mentioned as per your syllabus. Ibuprofen, this is the chemical structure of ibuprofen, and this you may observe that this is the propionic acid and connected that is placed at the one of the end of the benzene ring, and on the other the isobutyl side chain is present. So basically it is the 2-para-isobutyl propionic acid derivative, that is the ibuprofen. You may also memorize its name by the bu is representing the isobutyl. Clear? Para-isobutyl, ibu means isobutyl side chain, and profen, that is pro, is representing the propionic acid derivative. Next is the diclofenac acid. Diclofenac acid is contain the 2,6-ortho dichloro derivative in the phenyl acetic acid. So you have to only memorize the chemical structure of these ibuprofen and the diclofenac among the aryl, heteroaryl acetic acid, propionic acid, and NSAID derivative.